Bismillahirrahmanirrahim. This lecture regarding the embryonic period from 3rd to 8th week of development. As I mentioned in previous lectures, the pregnancy can be divided into 3 periods. Zygotic period from onset of fertilization to implantation, embryonic period from 3rd to 8th week, and fetal period from 9th up to 40 weeks. Regarding the embryonic periods, or regarding this lecture, the objectives are to describe the formation of neural tube, which is called neurulation, describe formation and fate of somites, also to describe the foldings of embryo, and identify the derivative of 3 gam layers: ectoderm, mesoderm, and endoderm. Starting with the embryonic period, it's a period from the 3rd to 8th week of development. Also, it's called organogenesis because in this period the organs initiate formation and start to develop. Also, at this time, each 3 games layer give a rise of a number of specific tissues, cells, and organs. The embryonic period is the most sensitive period, more sensitive to teratogens, and then it's called teratogenic period. Starting with derivatives of gamless, we will start with ectoderm derivatives. The ectoderm, as I mentioned, is the remnant of epiplast cell. In the 3rd week of the development, the notochord and perichordal mesoderm induce ectoderm to thickening. That area just above to the notochord and perichordal plate start to thick and form the neural plate. And the neural plate then is called neuroectoderm. While by the end of the 3rd week of development, this plate invaginates in the midline, and that start the formation of the neural groove, and then the thickening of the part of the neural groove to fold, and then later form the neural tube. The fusion of the neural plate or neural groove start at the level of the 5th somite in the cervical region and proceed or continue cranially and caudally to form the complete neural tube. This figure show here at the 18th day is a primitive streak, an area of primitive pit and knots, and beneath this region we have the notochord, and the notochord and perichordal mesoderm induce formation of the neural plate, and this is a neural plate. And then here groove appears in the midline, it's called neural groove, and then the groove and the plate together form the neural plate. Here we have the somites, and in this area the neural tube formed cranially and caudally. Also, this figure show section in the area of formation of neural tube. This is a notochord, this is a neural plate, and here we have the neural crest, and here neural fold and form neural groove. And then the neural crest cells detached from the neural groove, and the neural groove also each edge meet the other to form the neural tube. And finally, here we have the derivatives of the neural crest, and here we have the neural tube, and here this is the notochord. Regarding neurulation, or formation of neural tube and central nervous system, we have neural tube which has two ends open. The cranial neuropore or the cranial opening of the neural tube closed at day 25, and the caudal neuropore closed at day 27, and day 25 corresponding to 18 to 20 somites, and day 27 corresponding to developing of 25 somites. So the CNS or central nervous system presented by closed tube with a neurocaudal end that form spinal cord and broad cephalic portion which is called brain vesicles. This diagram show here this is somites, this is a neural tube, this is a neural fold, and as I mentioned before, the closure of neural tube starts from the level of fifth somite and then start cranially and caudally. Finally, we have the neural tube with anterior or cranial neuropore and caudal neuropore. Here is a figure at 25 or day of development. The connection of yolk sac is cut edge of amnion, the connecting sac, the posterior neuropore, the anterior neuropore, and here we have the somites and pharyngeal arches. At day 28 is the embryo, age 28 days. This is the umbilical cord, and here we have the caudal end and cranial end. Here we have the neural tube closed completely, just medially to the somites, and here we have the somites and limb bud or limb ridge which form the limbs later on, and here we have the pharyngeal arches and heart bulk, that the area of formation of the heart. So continue neurulation. When the neural tube is formed, detached from the surface ectoderm and just lies deep to it. During this detachment, some of these cells, inner border, fold, undergo epithelial to mesenchyme transition and detach from the neural tube and form neural crest mass. As I mentioned before, at the edge of the neural fold, we have the cells or a special type of cells called neural crest. During formation of the neural tube, the neural crest cell detach from the both ectoderm and neural tube, while the remaining ectoderm is called surface ectoderm, and that forms the epidermis of the skin. Here again is a notochord. We have here neural plate and neural groove, neural crest cells, starting here neural groove, and the neural tube start to form. Neural crest cells then detach from both neural tube and surface ectoderm. Here the surface ectoderm develop in epidermis, and the neural tube form central nervous system including spinal cord and brain, and here we have the neural crest cells or masses and their derivatives. So starting from here, the formation of the neural tube, which was plate and then folded there and form neural groove and start folding again and again to form the tube. Neural crest cells detach from the surface ectoderm and neural tube and form derivatives of it, including spinal root ganglion. The neural crest cells, it form from the cephalic region, migrate before closure of neural fold, and contribute to skull bones. Also cranial ganglia, glial cells, and melanocytes. While the neural crest from the trunk region leave the neural fold after closure of the tube and then migrate through one of two pathways, either dorsal pathway or ventral pathway. If it's migrate via dorsal pathway, so they migrate through the dermis and enter the epidermis and form melanocyte of the skin and hair follicles. If it migrates in ventral pathway, they migrate through the half of each somite, and the somite have a derivative of paraxial mesoderm. Then the ventral pathway of neural crest form the spinal sensory ganglia, sympathetic and enteric ganglia, Schwann cells, and adrenal medulla. This figure shows the neural crest cells, and then it detach from neural tube and neural ectoderm, and they form dorsal root ganglia, sympathetic ganglia, or adrenal gland or suprarenal gland, and pre-aortic ganglia, also enteric ganglia of GIT. Here, neural crest cells of the cranial region form the bones of the skull. The derivative of the neural crest cells, including connective tissue and bones of the face and skull, also cranial nerves ganglia, and cells of thyroid glands or C cells of thyroid gland, also conotruncal septum of the heart, also odontoplast, dermis in the face and neck, spinal root ganglia, sympathetic chain and pre-aortic ganglia, parasympathetic ganglia and gastrointestinal tract, adrenal medulla, Schwann cells, glial cells, arachnoid and pia mater, and melanocytes. Here, the ectoderm, which form the remaining part of epiplast, form the neural tube and neural crest cells and its derivative. The remaining part or other ectoderm give us the ectodermal thickening of otic and lens placodes, which later form the vestibulocochlear apparatus and lenses, which appear in the cephalic region. Also contribute to limb bridge that stimulate development of upper and lower limbs. Here this figure show the otic placode and lens placode, and here we have the limb bridge over there, and this around the 28th day of development. That is regarding the ectoderm. Regarding the mesoderm and its derivatives, as I mentioned before, the mesoderm or primitive mesoderm formed by migrating epiplastic cells through the primitive streak between the endoderm and epiplast cells, and it's arranged in three regions according to the notochord. Just lateral to notochord we have paraxial mesoderm, which is thickened. Lateral to paraxial mesoderm we have intermediate mesoderm. It's relatively thin comparing with paraxial mesoderm. A lateral plate mesoderm, and these small spaces appear in the lateral place and join to form the cavity, which is intracoelomic cavity or intra-embryonic coelom. And this divides the lateral plate into parietal and visceral layer. Parietal called somatic and visceral called splanchnic. So we have two lateral plate types of mesoderm. We have somatic and splanchnic, parietal and visceral layers. And here this diagram showing the three parts of mesoderm. Here we have the notochord, the neural plate, the neural groove, neural tube later on. And here we have in red color, this is the mesoderm. Just lateral to the notochord we have paraxial, and here we have intermediate, and here we have two layers of lateral plate mesoderm. Again, here we have the paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm with visceral layer and parietal layer, or somatic layer and splanchnic layer. Okay, the paraxial mesoderm is organized cephalocaudally and then appear in segments, and these segments called somitomeres. The somatomeres more loosely organized in the head region and forming in association with segmentation of the neural tube in into neuromeres. The somites itself is more compact and defined region forming from occipital region and caudally. The first somite appear at the 20th day of development, and the last pair appear at the end of the 5th week of development. Here, this is somites, and here we have the notochord. Here is the neural groove, the notochord, and here we have the ventral somite wall, and here we have the dorsal somite wall. The fate of somite: each somite can be divided into two main portions, sclerotome and dermomyotome. Sclerotome and dermomyotome. The sclerotome migrate around the notochord and spinal cord, forming the vertebral column. In addition, it forms the tendon for its muscle, the muscle around the vertebral column. The dermomyotome forms the limbs and body wall, musculature, muscles of the back, and the dermatome form the dermis and subcutaneous tissues. Here, this is the neural groove and notochord, and here you have the paraxial mesoderm and somites. Here's the ventral wall of somite, and it's arranged into sclerotome in this region and dermomyotome in this region. Here is a dermomyotome and sclerotome. So the dermomyotome then differentiated into myotome and dermatome surrounding the neural tube, while the sclerotome later on form the vertebral column that's surrounding the neural tube. The number of somites appear in the fetus with approximate age. It's formally used to identify or estimate the age of embryo. So from 1 to 4 somites corresponding to the 20 days, 21 day corresponding to 4 to 7 somites, 22 days corresponding to 7 to 10 somites, 23 around 10 to 13 somites appear, 24th day 13 to 17 somites, 25th day 17 to 20 somites, 26th day corresponding to 20 to 23 somites, 27th day around 23 to 26 somites, 28th day around 26 to 29 somites, and finally 30 days around 34 to 35 somites. That means additional 3 somites for every day from day 20 to day 28. That is regarding the paraxial mesoderm. Regarding intermediate mesoderm and lateral plate mesoderm, the intermediate mesoderm form the orogenital system, and the lateral plate of mesoderm give and divide into, as I mentioned before, into somatic and splanchnic layer. Somatic layer form parietal layer of the bodies. Splanchnic layer contribute to the wall of GIT and form visceral layer of it. Here, this figure showing the parietal mesoderm layer and visceral mesoderm layer. And here, this is the parietal layer of mesoderm, and this is the visceral layer of mesoderm. The blood and blood vessels formation. We have two ways to form blood vessels. The first way is vasculogenesis, whereby the vessels arise from the blood islands, or angiogenesis, and this is entails surrounding from existing vessels. While hemangioblasts are a common precursor of vessels and blood cells formation. The hemangioblast in the center of the blood islets or islands form hematopoietic stem cells. The peripheral part of hemangioblasts differentiate into angioblasts that precursor to blood vessels. So the source of blood and blood vessels are the hemangioblastic cells. And this figure shows the formation of blood and blood vessels. The mesenchymal cells. From mesenchymal cells, we have a blood islands here. Surrounding the center, the hemangioblasts differentiate to form the blood vessels, while in the center, we have the primitive blood cells, which formally has a nucleus to give a lot of number and types of blood cells. So finally, we have a lumen, which contain primitive blood cells and surrounding primitive blood vessels. All types and cells of blood and blood vessels arise from mesenchymal cells. Regarding this figure, shows the formation of blood and blood vessels surrounding the yolk sac and the area of chorion and syncytial trophoblast and cytotrophoblast as an event of functioning placenta. As I mentioned, formation of villi, primary and secondary and tertiary. The tertiary villi appear when the blood vessels and blood formed in the core of mesenchyme of the villi. Regarding the endoderm derivatives, the derivative of endoderm including GIT tract, a main organ system derived from endodermal layer. The epithelial lining of respiratory tract, thyroid gland, parathyroid gland, liver, pancreas, and epithelial lining urinary bladder. All these from endoderm. The main derivative is the gastrointestinal tract, completely derived from endodermal layer. The embryonic period also have an event of folding. The embryo formerly was a plate or disc, and then the folding of embryo make the embryo recognized as a human. We have three folds. We have a head fold and tail fold and lateral folds. Starting with the head fold, the trilaminar disc is folded into a 3D embryo that is recognizable as a human rather than a disc, with a cephalic or head fold, caudal or tail fold, and two lateral folds. All these stimulated by development of central nervous system and development of neural plate. Starting with the head fold, the growth of the forebrain beyond the buccopharyngeal membrane result in the head fold, which push the heart and pericardial coelom and septum transversum down and incorporates a portion of the yolk sac forming the foregut. In addition, into placing buccopharyngeal membrane at the sides of future mouth. Hereby, the head folds reorganize the buccopharyngeal membrane and cardiac region and initiate the formation of the foregut. And the foregut is a part of GIT formation. The GIT formed in three regions or three parts: foregut, midgut, and hindgut. The foregut here developed during the folding or head fold of embryo. This figure showing the folding of head portion or head fold. Here we have the septum transversum, amnion. Here we have the yolk sac here. Here we have oropharyngeal membrane. Here developing brain or forebrain. This is a notochord and neural tube here. So in this portion, the head fold occur ventrally like that and push the cardiac region from caudal portion to ventral portion. Here is a folding or head fold. So the developing mouth appear to be ventrally rather than cranially, and the cardiac region of the heart from ventral or from cranial become ventral. And this area later become the foregut. Regarding the tail fold, the primordial of spinal cord stimulate the tail fold, by which it puts the connecting stud to the ventral aspect of the embryo, incorporate with the portion of the yolk sac forming the hindgut. In addition, incorporating part of allantois to the body of the embryo, that shifting the position of cloacal membrane to side of the future anus. As what happened in head fold, here in tail fold also we have pushing up many structures to be ventrally, and that result in formation of hindgut. Also shifting of cloacal membrane to the side of future anus. This figure showing here the cloacal membrane. This is the neural tube and spinal cord area, the connecting stalk, and here we have the allantois. When folding ventrally or tail fold appear here. This is a amniotic cavity from dorsal become slightly ventrally. This is umbilical cord become ventrally, and here we have the allantois, and here this is the future hind gut, and this is the cloacal membrane presented in the side of future anus. So here this is the diagram showing the embryo from disc to two folds, head fold and tail fold, to become like that. It's folding from cranial part and caudal part. So the amnion become from dorsal, incorporate to the ventral part of the cranium and ventral part to the tail or caudal part of the embryo. So as a result of the head fold and tail fold, the heart become in the side, also the oropharyngeal membrane and the cloaca and allantois incorporate to the ventral part of the body, and formation of foregut and hindgut. The remaining part becomes the midgut. That's regarding the cavity which formerly was the yolk sac. So small part of yolk sac remain, while the most of it share information of GI tract or gastrointestinal tract as the foregut, midgut, and hind gut. Continue studying the folding of embryo. We have the lateral folds. Both of spinal cord and somite stimulate the lateral folding, which causes incorporate to another portion of the yolk sac, forming the midgut. And ventrolaterally of the body walls become ventry and fold, and the embryo now it can be recognized as a human embryo. The connective start with the yolk sac reduced to yolk sac stalk, or is called omphalonteric duct, which is the site of the future umbilicus. As we see here in this figure, the cranial and this is the lateral folding. This is a section or coronal section of the embryo, showing the two lateral folds. When folding there and there, the amniotic cavity also incorporate to the embryo from dorsal and become ventrally to the lateral part of the body, like that. And here we have the intra-embryonic cavity appear here. This is the yolk sac and connecting part of the yolk sac. And finally, here we have the gut and body cavities. And here we have the amniotic cavity surrounding all the embryo from all sides as a result of head fold, tail fold, and lateral fold. And finally, the human embryo can be recognized like this, with a head area and tail area, upper limb bud and lower limb bud, and this is the umbilical cord, and here we have the cardiac region, and this is the somites, and here beneath to this area we have the neural tube which forms the central nervous system. And this 5th week age embryo, showing the general features of the embryo at the 5th week. Also, we can see here 28 somites. You can see also the pharyngeal arches. And here this is the embryo. And guess what this? Yes, remnant part of yolk sac. And here, around 6 weeks age embryo, the yolk sac, the umbilical cord, here is the head region, this is the limbs, the cardiac region, and here we have the amnion surrounding the embryo from all sides. This is a placenta with chorionic villi and chorionic plate. Yeah, this figure of 7th week age embryo, the yolk sac, the chorionic cavity, and this is the amnion, the umbilical cord, upper limbs, lower limbs, eye placard, and here you can recognize the lenses of the eye. 7th to 8th week of the embryo, you can easily notice the head region and limbs there and there. Here we have the umbilical cord, and we can see here the size of yolk sac. Here we have the amnion, and here we have the chorion. So by the end of the embryonic period, the embryo can easily recognized as a human. All major organs are in place and startly formed, and then we can measure the length of the embryo and easily estimate the age of the embryo by crown rump length. The crown rump length This measurement used by ultrasound from the tip of head region to the tail region, and it can be used for determining the age of embryo by week. So from 5th to 8th millimeter length of crown rump, the age of embryo around 5th week. From 10 to 14, around 6 week age embryo. From 17 to 24 to 22, about 7th week of development. From 28 to 30, around the 8th week of development. That is regarding the embryonic period. In summary, the ectoderm give a rise to epidermis, a nervous system also responsible for forming skin, hairs, eyes. Mesoderm give a rise to skeleton, striated muscle, connective tissue, some part of skull and jaw. Mesoderm also source of blood cells and blood vessels that supply all the organs. Also forms the bone marrow, reproductive and excretory organs. The endoderm give the primary to epithelial tissue and lined digestive tube, lungs, liver, urinary tract, and germ cells. This figure shows the derivatives of the three germ layers. Here we have the derivative of ectoderm, including skin, central and peripheral nervous system. Here the derivatives of mesoderm, including muscles, heart, kidneys, bones, and so on. And these are the derivatives of endoderm, including the digestive tract or gastrointestinal tract, liver, and endothelium or epithelium of lungs and respiratory tract. Again, this diagram showing the derivatives of all germ layers, starting from blastocyst. The inner cell mass give the epiblast. From epiblast we have the three embryonic layers: mesoderm, ectoderm, and endoderm. The mesoderm in the head contribute to the dentine and connective tissue of the head and parts of skull. Paraxial mesoderm contribute to muscles of the head, striated muscles, trunk and limbs, skeleton except skull and dermis of skin, and connective tissue. Intermediate mesoderm give urogenital system including gonads, ducts and accessory glands. Lateral plate mesoderm give a connective tissue and muscles of the viscera, serous membrane, pleura, pericardium, peritoneum, primitive heart, blood and lymph cells, spleen, suprarenal gland. While ectoderm, the surface ectoderm give a rise to lens of the eye, internal ear, enema of the teeth, anterior part of pituitary gland, epidermis, hair, nails, cutaneous and mammary glands. Also give anterior part of pituitary gland. Neuroectoderm, the neural tube contributes central nervous system, retina, pineal body, and posterior part of pituitary gland. The neural crest cells give a rise to cranial accessory ganglion nerve, medulla of suprarenal gland or adrenal gland, ligament cells or melanocyte, pharyngeal arch cartilage, head mesenchyme and connective tissue, bulbar and conal ridge of the heart. While the endoderm gave a rise of epithelium of gastrointestinal tract, liver, pancreas, urinary bladder, and urachus. Also, epithelial part of the pharynx, thyroid gland, tympanic cavity, pharyngeotympanic tube, tonsils, and parathyroid glands. For any questions or any points in this lecture need clarifications, just send me on Telegram. I'm going to answer you. Thank you and good luck.